At my conception, each of my parents' gametes contributed 23 molecules of DNA, known as chromosomes. The gene known as BRCA1, which is very important in preventing cancerous cell growth and preventing breast cancer specifically, is located on 17q21, the long arm of chromosome 17. At a specific spot in the BRCA1 gene, at nucleotide 3726, I changed the normal cytosine-guanine base pair that most people possess to an adenine-thymine base pair so that the cytosine was replaced by a thymine. This change in the DNA nucleotide sequence resulted in a change in the mRNA transcript sequence, so that a codon which reads CGA in most individuals is instead UGA when my mRNA transcripts are produced. Most individuals produce a BRCA1 protein which is about 1,800 amino acids long, and at residue 1,203, the amino acid arginine is inserted. However, because of my abnormal codons, this residue is not inserted. In fact, UGA is a termination or stop codon, and translation stops here, and about 600 amino acids are not inserted. My BRCA1 proteins are only about 2/3 the normal length. In normal cells, BRCA1 prevents cells from dividing inappropriately and thus helps to prevent cancer. In my cells, the truncated protein does not function normally, and a potentially cancerous cell may divide when it shouldn't. BRCA1 is a very important protein which can interact with a number of regulatory proteins in the cell and can bind to DNA. Because my BRCA1 protein does not produce many of the regions which interact with other proteins, it is non-functional, and therefore it is incapable of arresting cell division in certain cells which should not divide, and this was a factor in my developing breast cancer.